everyone, my name is Sarah, and today we're going to be talking about a topic that is a little bit more difficult than usual. I think maybe I'm just biased when it comes to the medical sciences, but today we're going to be talking about a new method for diagnosing cancer in the early stages. So obviously very, very important research that is being done, also very, very complicated, so bear with me here as we make our way through this slightly, medium challenging article. And if you see any words that you don't know, do not hesitate to stop and look them up or try to listen to my English explanations. But again, a lot of these words are really difficult even for English native speakers because it's a lot of scientific jargon. So yeah, don't feel bad if you don't recognize all of the words. But hopefully we can get through this together. All right, let's take a crack at it. Let's try. The results of a study performed by the University of California at Irvine have demonstrated the potential effectiveness of using a novel method of biomarker identification for early stage cancer detection. Whew! Already in just that first sentence, we got 5 vocabulary words. Great. So first, I just want to point out this set phrase right here, to perform a study. This is constantly used in anything that's about any kind of academic literature, anything that was published in an academic journal. When you perform a study, it means that you do an experiment or you do a research investigation. So perform here, it does not literally mean to sing or dance or act, which is the other sense that the word perform is often used in. But yeah, so to perform a study, or a study was performed, just know that that means that research was done or they did some kind of experiment. Good to know. All right, so this study that was performed or carried out by the University of California demonstrated or showed us the potential effectiveness of using a novel or new method of biomarker identification for early stage cancer detection. So last vocabulary word in this sentence is potential. So in Korean, it's 잠재적인. So potential means that it might be effective. We don't know 100% for sure, but we know that it's like 잠재적인 효과성, potential effectiveness. So yeah, it's a little bit of a complicated expression to explain just in English, but yeah, just so you know, it's 잠재적인 효과성 in Korean. Great, and then of course, early stage is 초기, so early time cancer detection, which is obviously very, very important. Although the experiment primarily involved patients with lung cancer, preliminary results have suggested that the new approach could also be effective for treatment of conditions like Alzheimer's and other aging-related diseases. Great, so the experiment primarily, or mainly, involved patients with lung cancer. So this means here that most of the people who participated in the study had lung cancer, but not all of them. But it was definitely the majority, or more than half. So even though most of the people who were in this experiment had lung cancer, it was primarily involved with patients with lung cancer, preliminary or early results suggest that the new approach or method could be effective for treatment of conditions like Alzheimer's. And Alzheimer's, I believe in Korean, is 치매. So it's a disease that affects the brain, typically in people who are older, and it often results in people forgetting a lot of their memories, and it's very sad, and a lot of times the people are very upset and confused because they don't know where they are, they don't know who they are, they don't even recognize their own children or their spouse in some cases. It's a very, very sad disease. So anything that we can do to diagnose and treat diseases like Alzheimer's in the early stages is a really good thing. And that's why this study performed by the University of California at Irvine is important. Great. The new method, referred to as cell heterogeneity adjusted clonal methylation, or CHALM, we're just going to call it CHALM from now on, in the paper, improves the prediction of gene expression and biomarkers to help researchers and physicians more rapidly and reliably identify differentially methylated genes necessary for cancer diagnosis. Okay, let's count. 1, 2, 3, 4, 5, 6, 7, 8. Oh my goodness. 8 vocabulary words in just one sentence. That is a lot of vocabulary words. Okay, so first of all, CHALM, it's a 줄임말 or a abbreviation for cell heterogeneity adjusted clonal methylation. And if you don't know what a lot of those words are, you're not alone. I bet that 90% of Americans would look at that and be like, what? So, you know, if you don't know what that means, it's okay. You're not stupid. It's a really difficult concept. So, okay, this word here, cell, in Korean is 세포, right? Easy enough. Heterogeneity. So when something is hetero, it means it's different. And gene, so it's different gene-ness. So the diversity of genes in cells, cell heterogeneity, adjusted clonal methylation. So basically, they methylate or they inject or mix something with methanol, which is a substance or a chemical, based on its cell heterogeneity. So it's adjusted for cell heterogeneity. And yeah, if you don't know what that means, basically just know it's got something to do with cells, it's got something to do with methanol. They do some, you know, fancy science stuff on the cells so that we can predict gene expression and, you know, more accurately and quickly identify diseases. Great. All right, so let's just call it CHALM from now on. I know it's a little bit of a difficult concept. Yeah, literally, unless you're a cancer researcher, you don't actually need to know exactly the precise term for this, so like don't stress yourself out too much. Okay, so this new method, which we're just going to call CHALM, we're not even going to look at this again, it improves the prediction of gene expression and biomarkers. So when you predict something, you guess what is going to happen in the future, or you guess something that you don't know for certain yet. So this method, CHALM, it predicts gene expression. So how do genes in our DNA actually express themselves in a physical way inside of our bodies and, you know, in our appearances? It predicts the expression of these genes as well as biomarkers. And so this word biomarker, it's an indication that is, it can be identified using biology. So biology is 생물학, it's the life sciences in English, and marker, so it's an indicator that is biologically evident. So it's something that the scientists can see either physically in the human body or if they look really close at our cells and our DNA. So these biomarkers are going to be on our body in various different ways. Okay, so CHALM, it's used to guess or predict gene expression and biomarkers. And this helps the researchers rapidly, that means quickly, identify differentially methylated genes necessary for cancer diagnosis. So as we mentioned before, the word methylate, it means to mix or inject with methanol, which is a chemical substance. And seeing how these methylated genes are different from each other and how much methanol they have in them based on their differences, so differentially methylated, you can identify or find biomarkers that are necessary or needed for cancer diagnosis. And a diagnosis is when a doctor says basically like what kind of illness it is that you have. So you need to have a diagnosis before you can get a treatment. You need to know that you have cancer before you can get treatment for cancer, right? So when the doctor says you have cancer, that's called a diagnosis. Of course, I wouldn't wish that on anybody, but yeah. Okay, so just one more time, we're going to read the sentence just one more time. I know it's really difficult, just hang on with me. We're going to be okay. So this new method done by the scientists at University of California, Irvine, called CHALM, it improves the guessing of gene expression and biomarkers, And this helps researchers quickly identify differently methylated genes which are needed to diagnose cancer. Okay? All right, just let it sink into your brain. Pause the video if you have to. And after you're done, we'll move on to the next paragraph. Let's take a look. While traditional methods have been limited in their efficacy by the finding that correlation between gene expression and promoter methylation is weak at best, the new research found that the CHALM method was able to better detect and analyze methylated markers than traditional methods. Very interesting. So this new method, it's called CHALM, and it's new because it's different from what is old or established or traditional. So traditional, it basically just means whatever was done the old way or the established way that most people do it. So for instance, in Korean culture, it's traditional to celebrate Seollal or the Lunar New Year with your family. But it doesn't literally have to do with like cultural traditions. It can also be a tradition in the scientists if it's an established practice that most people follow that has not changed in a while. So traditional methods are limited in their efficacy or how effective they are, or 효과성, by the finding that the correlation or 연관성 or relationship between gene expression and promoter methylation is weak. The new research found that the CHALM method better detects and analyzes markers. Okay, so here is another expression. So in scientific journal articles and newspaper articles about science, you will very, very often see this expression right here. The correlation between blank and blank is strong or it's weak. So for instance, the correlation between childhood nutrition and adult height, so how tall you are when you become an adult, is strong. Because usually if you're eating healthy things when you're young, you grow tall and healthy. And if you're unable to access those resources, you're going to end up being a few inches shorter than you might have been had you had access to the proper nutrition as a child. So when we say that the correlation between gene expression and promoter methylation is weak, it means that basically this traditional method that we've been using, that most people are using, and it's the established way, it's actually not that good at predicting what it's supposed to predict. Because basically if the relationship is not linear, if it's not something that you could really predict, if it's just kind of random, then the test that you're using is actually not very effective at all. So in this situation, the correlation is weak, but what we want is something where the correlation is strong. So these two variables, they follow each other in a predictable way that makes it more easy for researchers to guess what is going to happen and hopefully help people diagnose their cancer and their other health-related issues earlier so they can get more treatment. Whew. All right. So because this method, the traditional method, is limited by its weak correlation between the two variables that it measures, the CHALM method was able to better detect or find and analyze or 분석하다 methylated markers than traditional methods. So basically, this new method is better at what the traditional method is supposed to be doing, which is a good thing because that means we can make more accurate predictions and more accurate diagnoses and treatments. Whew. So who is responsible for all of this? Let's see. Wei Li, a professor at the UC Irvine School of Medicine and one of the scientists that worked on the study, explained his findings to the press after his research was published in the prestigious journal Nature Communications. Okay, so this one's a little bit easier, thank God. So here's just another expression to be aware of if you're reading any kind of newspaper articles about scientific journals or just reading the journals themselves. This is a set phrase to explain blah 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 to the press. And so the press in this case means the media or journalists or people who are going to make news articles or news videos that people consume later. And he explained his findings to the press or to journalists after his research was published in the prestigious or very famous, very high-ranking journal, Nature Communications. Okay, so this one has a lot more difficult words that I honestly did not know before I was researching for this video. So yeah, if you see something that is difficult for you, don't be afraid to just pause the video and look it up, because it's difficult stuff, unless you're a cancer researcher, which you're probably in high school, so I doubt that you're a cancer researcher. Anyway, according to his faculty profile, his lab is focused on the design and application of bioinformatics algorithms to elucidate global epigenetic mechanisms in neural development in diseases such as cancer. His other areas of expertise include DNA methylation, epigenetic regulation, alternative polyadenylation, and nucleosome organization. I literally did not know what the last half of the sentence meant before I did this video. He received his doctorate from the Chinese Academy of Sciences in 2003. Whew, a nice, easy sentence. Great. Okay, so we talked about Wei Li. He is one of the scientists that worked on the study. So we want to know, what kind of guy is he? Why should we trust the things that he says? So according to his faculty profile, so a faculty is someone who is a professor or a researcher at a university or some kind of educational institution. So there's all types of different people that can work at a school. So there are employees, which include faculty, but it also includes staff. So the staff are the people who are running the front desk. They're the librarians. They're the ones who are kind of making the logistical side of the university or the educational institution flow smoothly. But the faculty are the people who are actually doing the research and the teaching that is kind of like the core of the mission of these institutions. So he has a faculty profile, which means he's a professor or a researcher at UC Irvine. And according to this profile, it says that his lab is focused on the design and application of bioinformatics algorithms. So that means that they make, design, and apply bioinformatics algorithms. So bioinformatics algorithms is basically it's a type of computer code that can kind of talk to itself and make decisions on its own based on information from biology. So biological information. So you feed the biological information into the algorithm, and after you do that, you can elucidate or explain global epigenetic mechanisms in neural development and diseases such as cancer. So epigenetic, this is not a word that you need to know in normal everyday English, but basically it just means epigenetic. It means from basically from outside of the genes. So it's not the genes themselves, even though the genes are affected by it, but it's not the genes that are causing the issue, if that makes sense. Okay, so we looked at his faculty profile, and his lab makes and uses biological information, puts it into algorithms to explain epigenetic mechanisms in diseases such as cancer. Whew. Okay, so the very last sentence, let's make it to the end. You got this. So what else is he good at? His other areas of expertise, so things in which he is an expert or 전문성, include DNA methylation, epigenetic regulation, alternative polyadenylation, and nucleosome organization. You don't have to know exactly what these terms mean, but basically he is an expert on DNA, genetic material, and like the interplay that they have with the development of diseases. And how did he get so smart? How did he get to know not only what these words mean, but how to use them in a useful way in order to design experiments and, solve problems like cancer? Well, he received his doctorate, or his PhD, or his 박사, from the Chinese Academy of Sciences in 2003. Whew, I feel like I just got a doctorate in cancer research just reading this article. So I hope that all of you aspiring doctors out there got something out of reading this article. I know that this week's article is really, really difficult compared to some of the stuff that we normally do, so I am very, very grateful for your patience watching this video all the way to the end. All right, well, if you guys have any questions, comments, or suggestions, as always, you can let me know in the comment section below, and I look forward to seeing you guys next time. All right, have a great day. Bye.